A very good morning all the students. Today we will discuss the second class from the cytotoxic agent, that is antimetabolite. In the last lecture we have completed all the points related to the anti-neoplastic agents, right? We have completed the introductory part, some historical part, some rationale for the development of the drugs for the cancer. Again we have also discussed about the first class of the from the classification we have completed the first class, that is cytotoxic agent. From that the first class, that is alkylating agent. So the second class is the antimetabolite. We have completed with alkylating agent. Okay, now we will start with the next class, that is the antimetabolites. The antimetabolites are basically again classified into the three different classes. The first one is the purine antagonist, pyrimidine antagonist, and a folate antagonist. So these antimetabolites are classified into these three classes. Okay, so basically the name indicates that what is the name here? We have a antimetabolites. So what it indicates? It indicates it forms a metabolic product. So these are the compounds which form some metabolic products that biosynthesized or biological important products are formed, and these metabolic products are become toxic to the cell. Okay, so this is in simple we can say the mechanism of action of this drug. That means they form some faulty metabolic product during the DNA and RNA synthesis, and that faulty product will be utilized for the RNA and DNA synthesis, and which forms a faulty synthesis. Okay, or synthesis of DNA RNA does not be taken place, or further protein synthesis also inhibited, and again the cell division will not be takes place. So these drugs are basically act at the S phase if we consider the cell life cycle. Okay, so these antimetabolites are the compounds which are related in the structure to a cellular precursor molecule. So for the study of this drug we need to study how the synthesis of DNA and how there is a synthesis of RNA taken place, okay, in a particular cell. So what are the different mechanisms for the biosynthesis of DNA and RNA? Again, various proteins in our body, purine and pyrimidine analogues in our body. So that is very important. For the particular synthesis of purine and pyrimidine, the particular precursor molecules are required. These antimetabolites, these are the substances which are closely related in their structure with that of the precursor molecule. And these substances are capable of preventing the proper use or formation of normal cellular product. These antimetabolites are similar enough in their structure in many cases to interact with the normal cellular process, but they are differ in the manner sufficient to alter the outcome of that pathway. So what is they say? These are the antimetabolites which are similar in their structure also and interact with the normal cellular processes which are observed in a particular normal cell, but because of their differ in manner of formation of new product, they alter the outcome of that pathway. So because of the altering the outcome of that pathway, the pathway will not be completed in a normal way. So there is some disruption will be takes place in the particular pathway, and because of that cell death will be observed. Okay, so most of the antimetabolites are effective in cancer chemotherapeutic agents via interaction with the biosynthesis of nucleic acids. So therefore several of the useful drugs used in antimetabolite therapy are purine, pyrimidine, folates and related compounds. So I have told you the classification of this drug of this class. They are classified as a purine antagonist, pyrimidine antagonist and folate antagonist. Okay, so according to the useful drugs. Okay, these useful drugs are given in the therapy by purine, pyrimidine or folate. So they are classified into the three classes: purine antagonist, pyrimidine antagonist and folate antagonist. So the antimetabolite drug may exert their effect by several individual mechanisms involving enzyme inhibition. Okay, how they act? They may act by involving in the enzyme inhibition at the active, allosteric or related sites. Most of these drugs, these targeted enzymes and processes are involved in the regulatory step of the cell division and the cell or the tissue growth. Okay, so most of the time what happens, the targeted enzymes means what? The enzyme for which your antimetabolite will inhibit that their action. So these are the various enzymes which are targeted by the antimetabolite. They are involved in the various regulatory processes of cell division and a cell or tissue growth. Often the administered drug is actually a prodrug form of an antimetabolite and it requires activation in vivo to yield the active inhibitor. So sometimes these drugs are the act as a prodrug form. So their activation, so they need their activation whenever they enter in our body. So this is one of the part, and that activation basically carried out by the phosphorylation only. The administration of many purine and pyrimidine antimetabolites requires the formation of nucleoside and finally the corresponding nucleotide and metabolite for antimetabolite activity. So what are the purine and pyrimidine antimetabolites do? They requires the formation of nucleoside and finally corresponding nucleotide for antimetabolite activity. An antimetabolite and its transformation products may inhibit the several different enzymes which are involved in the tissue growth. So that antimetabolite is formed and their transformation product, both they may inhibit the several different enzymes which are involved in the tissue growth of the tissue or growth of the cell. These substances are generally a cell cycle specific with the activity in the synthetic phase. Just now I have told you that these drugs basically act at the S phase, that is they become a cell cycle specific drug. Okay, so next, the purine and pyrimidine antimetabolites are often compound incorporated into a nucleic acid and nucleic acid polymers. Okay, you know it very well. There are the two purine bases and two pyrimidine bases, right? These purine and pyrimidine bases are helpful for the DNA and RNA synthesis. They forms the nucleic acid and after that they forms a nucleic acid polymer, that is DNA and RNA, by cross linking with the hydrogen bonds, two double hydrogen or triple hydrogen bond. Okay, ACGT. So they form, these are the purine and pyrimidine bases only. Okay, so you know that. Just write down in the comment, or you just write down what are the purine bases and what are the pyrimidine bases in your notebook. Okay, so you get an idea. Okay, what are the purine or pyrimidine bases. So these purine and pyrimidine antimetabolites are incorporated into the nucleic acid and the nucleic acid polymers, that is DNA and RNA. The antifolates, so what are the purine and pyrimidine, that we get, but about folate. So antifolate are the compounds which are designed to interfere at the cofactor site for enzymes which are involved in the biosynthesis of nucleic acid bases. The biosynthesis of these nucleic acid bases depend on availability of folate cofactors. Hence, antimetabolites of the folates find utility in antineoplastic agent. So what is the use of antifolate drug? These drugs interact with the cofactor site for the enzyme, and that enzyme, what is the function of that enzyme? That function is the biosynthesis of the nucleic acid bases. So because of interference of these antifolate with that of the cofactors, the biosynthesis of this nucleic acid will be interrupted, and the cell growth will not be taken place. In this way this drug is act. So the next, the anti-tumor effect of all these compounds attempting to masquerade as the natural precursor compound may occur because of the malfunction in the biosynthesis of the corresponding macromolecules. The classic examples for these purine and pyrimidine antimetabolites are 5-fluorouracil, 6-mercaptopurine, and classic example for antifolate is methotrexate. So these are the three different examples. You just remember all these examples. Okay, 5-fluorouracil, 6-mercaptopurine, and methotrexate. The 5-fluorouracil is a pyrimidine antimetabolite, while 6-mercaptopurine is purine antimetabolite. Okay. The purine analog 6-mercaptopurine and its bioactivation products interact with many enzymes in various stages of the cell division. So the activity of the 6-mercaptopurine requires activation. The activation of 6-mercaptopurine requires bioactivation to its ribonucleotide 6-thioinosine by the enzyme hypoxanthine guanine phosphoribosyl transferase. So the purine analog is 6-mercaptopurine. Its bioactivation product interact with the many of the enzymes at various stages in the cell division. The activity of 6-mercaptopurine requires the bioactivation to its ribonucleotide 6-thioinosine by the enzyme hypoxanthine guanine phosphoribosyl transferase. The 6-thioinosine is a potent inhibitor of the conversion of 5-phosphoribosyl pyrophosphate into a 5-phosphoribosyl amine. So this is one of the inhibitor, that is 6-thioinosine is a drug which is the inhibitor for the conversion of 5-phosphoribosyl pyrophosphate into a 5-phosphoribosyl amine. The ribose, diphosphate and triphosphate of 6-mercaptopurine are active enzyme inhibitors, and the triphosphate can be incorporated into the DNA and the RNA to inhibit the chain elongation. So in this way these 6-mercaptopurine are act. That is 6-thioinosine is an inhibitor for the conversion of 5-phosphoribosyl pyrophosphate into the 5-phosphoribosyl amine. The ribose, diphosphate and triphosphates of 6-mercaptopurine are active enzyme inhibitors, and the triphosphates can be incorporated into the DNA and RNA, and which then inhibit the chain elongation. Okay. So in this way these drugs show their pharmacological action, and the cell will be cell death will be taken place. The pyrimidine analog, that is 5-fluorouracil, was designed based on the conversion in the certain tumors. Uracil can be used for the nucleic acid pyrimidine synthesis. Fluorine at the position 5 of the uracil blocks the conversion of uridylate to thymidate, thus diminishing the DNA biosynthesis. The fluorine which is there in the 5-fluorouracil increases the acidity, that is inductive effect, and was expected to cause the molecule to bind more strongly to the enzymes of the various stages of pyrimidine biosynthesis. The 5-fluorouracil is one which having a 5-fluoro group on the uracil molecule. So it's a pyrimidine analog which is used in the treatment of various certain types of the tumor. It is involved in the biosynthesis of thymidine nucleic acid. Okay, so fluorine at the position 5, it has a fluorine group. It blocks the conversion of uridylate to thymidylate, diminishes the DNA biosynthesis. So in this way it act as the inhibition of the DNA biosynthesis, and further cell growth and all the steps of the cell division are inhibited. The fluorine induced increase in acidity, that means inductive effect, and it is expected to cause the molecule to bind more strongly to the enzymes of the various stages of pyrimidine biosynthesis. So this thymidine is a logical target because it is only found in the DNA molecule and this is not observed in the RNA. The 5-fluorouracil is activated by the anabolism 2,5-fluoro-2-deoxyuric acid, and this activated species is strong inhibitor of thymidylate synthetase. The enzyme that converts 2-deoxyuridylic acid to the thymidyllic acid. So got the mechanism of action of this 5-fluorouracil. This 5-fluorouracil is involved in the biosynthetic step, okay, of the DNA. Okay, so this basically form 5-fluoro-2-deoxyuridylic acid. Okay, and that 5-fluoro-2-deoxyuridylic acid inhibit the enzyme thymidylate synthetase. And what is the function of that enzyme? That function of that enzyme is to convert 2-deoxyuridylic acid into a thymidyllic acid. So that conversion will not be takes place. Again, next, the methotrexate. This inhibit the binding of the substrate folic acid to the enzyme dihydrofolate reductase, resulting in the reductions in the synthesis of the nucleic acid bases and conversion of uridylate to thymidylate, which is catalyzed by thymidylate synthetase, which requires folate cofactors. Okay, so this methotrexate inhibit the binding of the folic acid to the enzyme dihydrofolate reductase. So instead of going for the reduction, this drug shows the reduction in the nucleic acid bases, and also it's involved in the conversion of uridylate to thymidylate. Okay, and this conversion requires some folate cofactors, that is dihydrofolate, tetrahydrofolate, okay, that are related to the that is required for the required as a cofactors. So that cofactors will not be synthesized. So the biosynthesis of pyrimidine and purine monomers is a central component for the DNA and RNA formation in the mammalian cells. The process of this cell division and tissue growth is very complex set of biochemical events with many feedback controls. Thus, antimetabolite drugs based on the pyrimidine, purine and folate and related structures may exert their effects by altering many metabolic processes as well as metabolic pathways. So the final step in illustration illustrates the conversion of ribonucleotide to the deoxyribonucleotides catalyzed by the ribonucleotide reductase enzyme. So this final step is also an important step, right? The substrate for this enzyme is ribonucleotide diphosphate, and the process is same for the both purine as well as pyrimidine bases. So this is the important point related to the cell division or the cell growth, okay, or the various pathways or the processes involved in the cell. So how this pyrimidine drug shows their action and how they affect the DNA and DNA synthesis. Okay, the pyrimidine derivatives, that is 5-fluorouracil, is involved in the, what we have said, that is a drug. What is the action? That is involved in the, or the blocks the conversion of uridine to thymidine, right? So the normal thymidine involves methylation at the 5th position of the pyrimidine ring of the uridine. So what is thymidine? Thymidine is the uridine. How uridine, when uridine is converted into thymidine, what happens? At the 5th position of this uridine is get methylated. Okay, so at 5th position we have a fluoro group. So in case even there is a uracil is there or uridine is there, in uridine it has a hydrogen, and that hydrogen will easily replaced with the methyl group and it convert to the thymidine. Okay, but what happened, because of the presence of the fluorine at the 5th position, this drug act as a antimetabolite drug, which results to the formation of the stable covalent ternary complex, that is complex composed of 5-fluorouracil, thymidylate synthase and cofactors. So this replacement of the hydrogen at this 5th position of the uracil with an fluorine, which results in an antimetabolite drug, which leads to formation of the stable covalent ternary complex composed of 5-fluorouracil, thymidylate synthase and the various cofactors, that is tetrahydrofolate. The normal pathway for the formation of thymidine from the uridine, which is catalyzed by the enzyme thymidylate synthase. Okay, what is TS? It's a thymidylate synthase, is shown in this scheme. Okay, the anti-cancer drug targeting this enzyme should selectively inhibit the formation of DNA, because the thymidine is not a common normal component of the RNA. So this drug, that is pyrimidine drug, which inhibits the synthesis of DNA only. So what is the scheme? We just see the scheme. So what they have mentioned here, the normal pathway. Normally how this thymidine is obtained from the uridine. Okay, so uridine, arrow, thymidine, on the arrow it requires the enzyme, that is thymidylate synthase. Okay, so this is the scheme for the biochemical conversion of uridine to the thymidine. So what is the role of this thymidylate synthase enzyme? deoxyuridine monophosphate, okay, with the help of this enzyme, and this cofactor is required, that is tetrahydrofolate cofactor is required. So with this drug is converted into this complex molecule, okay. That complex then undergo the methylation at the 5th position. Here again this methylation, then again this thymidylate synthase is released as such. So here we have a methyl group at the 5th position. Again that tetrahydrofolate factor is there, which is converted into a dihydrofolate. Okay, so this is the step for the biochemical conversion of uridine to thymidine. Just I will repeat the explanation again. So what is this? This is deoxyuridine monophosphate. You may consider it as the uridine only. Okay, this is a uridine. So in the uridine, what is the substrate? Is the uridine. This is the enzyme required, that is thymidylate synthase, and this is the cofactor required. Okay, what is the role of that cofactor? To add the methyl group. Okay, that is the tetrahydrofolate folate enzyme is there, cofactor is there, which is responsible for addition of or replacement of this hydrogen with the methyl group. Okay, so this step we need. So we have this complex molecule is formed. Okay, after formation of this complex molecule, there will be the formation of this CH2, double bond CH2 methylene, and that methylene group is converted into the like this, that CH3. So accordingly that tetrahydrofolate is converted into dihydrofolate. That enzyme remain as such, it just acts as the enzyme for the conversion, okay, for addition of these two molecules. Okay, and then here we have a thymidine molecule, that is this is thymidine. So the conversion of uridine to thymidine, it requires an enzyme thymidylate synthase and tetrahydrofolate. So next, the thymidine synthase is responsible for the reductive methylation of deoxyuridine monophosphate by 5-10 methylene tetrahydrofolate to yield deoxythymidine monophosphate and dihydrofolate. Because thymine is unique to the DNA, the thymidine synthase enzyme system plays an important role in the replication and a cell division. The tetrahydrofolate is a cofactor species which serves as both the carbon donor and the hydride source in this system. So just you got an idea now. So it's next here. The initial step of the process involves the nucleophilic attack by the sulfhydryl group of the cysteine residue at the 6th position of the deoxyuridine monophosphate. The resulting enolate adds to the methylene of 5-10 CH2THF, that is tetrahydrofolate, perhaps activated via a very reactive N5 iminium ion. This iminium ion is forms at N5 and only after the use of this cofactor, that is 5-10 tetrahydrofolate, and binds to the thymidylate synthase. Okay, so this is the important and steps, and what happened or what should not be happened. Okay, so which steps is very important, and when this step will not be happened, so how the further steps or the further pathways will be disrupted. So iminium ion is likely formed at N5 because it is more basic to the two nitrogens, whereas N10 is better leaving group. The loss of proton at the fifth position of the deoxyuridine monophosphate, and elimination of the folate yields the exocyclic methylene uracil species. The final step involved hydride transfer from the tetrahydrofolate and elimination to yield the enzyme dihydrofolate and deoxythymidine monophosphate. The 5-fluorouracil is activated by the conversion to a corresponding nucleotide species, that is 5-fluoro-2-deoxyuridylic acid. So this is the 5-fluorouracil which is converted into a or which is activated first. After then it is activated triphosphate, it interfere with the DNA, and when it's activated and forms a 5-fluoro-2-deoxyuridylic acid or deoxyuridylic monophosphate, then it forms a triphosphate and it interferes with the DNA. So in this way this mechanism of action is observed. The 5-fluoro-2-deoxyuridylic acid is powerful inhibitor of thymidylate synthetase, the enzyme that converts 2-deoxyuridylic acid to the thymidyllic acid. In the inhibition reaction, the sulfhydryl group of this thymidine synthase adds via conjugate addition to the sixth position of the fluorouracil moiety. The carbon at the 5 position then binds to the methylene group and of the 5-10 methylene tetrahydrofolate following initial formation of the more electrophilic form of the folate, the N5 iminium ion. In the normal process, this step is followed by the elimination of dihydrofolate from the ternary complex, the regeneration of the active enzyme species, and the product thymidine is obtained. So because of this 5-fluorouracil, this form, it forms the 5-fluoro-2-deoxyuridylic acid. This is binds with the thymidine synthase, and this thymidylate synthase will not be released for further action. So it shows the inhibition action at the thymidylate synthetase enzyme. So the central to this process is the loss of the proton at the fifth position of the uracil to form the exocyclic methylene uracil species. The 5-fluorine is stable to the elimination and the terminal product will be result involving the enzyme cofactor and substrate, all covalently bonded together. The chemical mechanism of inhibition of thymidylate synthetase by the 5-fluorouracil is shown in this scheme. How this 5-fluorouracil is inhibit the thymidylate synthetase. So this is the tetrahydrofolate cofactor. Again this is the 5-fluorouracil. This is the enzyme. So this is the very stable complex. Here we have a fluoro group. Okay, this fluoro, and this ternary complex which is formed, it's a very stable complex which does not undergo further conversion. Okay, because of that what happened, that thymidylate synthase enzyme will not be released. Again this factor which is there, that is dihydrofolate, is also not released, and thymidine will not be synthesized. Okay, so this process clearly shows that in order to inactivate the thymidylate synthetase enzyme, the both 5-fluorouracil and the tetrahydrofolate species are required to form a ternary complex. Some clinical studies have shown that the administration of the tetrahydrofolate source prior to the treatment of 5-fluorouracil results in greater inhibition of total thymidylate synthesis activity. The administered source of active 5-10 methylene tetrahydrofolate is a leucovorin N5-formyl tetrahydrofolate. The thymidylate synthetase is the most obvious and well documented mechanism of action for the 5-fluorouracil, which is having a cytotoxic action. However, other mechanisms may play a role in the overall value of this drug in the treatment of human cancer. So the triphosphate of this 5-fluorouracil is also shows the particular action. Okay, so various anti-cancer drugs which are having or which are from the pyrimidine nucleic acids, that is the examples are 5-fluorouracil. Second example is floxuridine, capecitabine, tigafur, gemcitabine, citarabine, 5-azacitidine, decitabine. So these are the different drugs, different anti-cancer drugs from which are of the pyrimidine analogs. Okay. So next, these pyrimidine analogs, these are act as the antimetabolite for the cancer therapy. They have been developed based on the cytosine structure as well. The modification of normal ribose to deoxyribose moiety has produced useful drug species such as citarabine, that is Ara-C, and the gemcitabine, the cytosine arabinoside, that is Ara-C or citarabine is simply a arabinose sugar. So this drug also involved, this is modified drug, okay, and this drug also involved in the previous stages of this DNA of the cell division, that means formation of the sugars. Okay, deoxyribose sugar we have to synthesize. Okay, that sugar molecule come together with the phosphate, then it forms a binucleotide, then trinucleotide and all. Okay, so the anti-cancer drugs which are based on the purine are and their related compounds. Example is 6-mercaptopurine. Just draw the structure of the purine. At 6th position we have to add the mercapto group, that is SH, thioguanine. So only the difference is the addition of this amino group here, thioguanine. Then cladribine, vidarabine, clofarabine, fludarabine, pentostatin. So these are the different drugs which are the purine analog. Next is the folate. Methotrexate is the basic or the classic example for the folic acid antimetabolite, which is structurally derived by N-methylation of the para-amino benzoic acid residue of the PABA and replacement of the pyridine hydroxyl by the bioisosteric amino group. See here, this is the structure of folic acid. Okay, so this folic acid contains, this is the glutaric acid, this is the amino acid. Okay, this is glutaric acid, CH2, CH, COH, NH2, CH2, CH2, COH. This is the glutaric acid, and this is the para amino benzoic acid, NH2, C6H5, COOH. This is para amino benzoic acid, and this is your pyridine structure. So this is the structure of folic acid, and this is the structure for methotrexate. So what is the difference you see here? First difference is the methylation. Okay, methylation, N-methylation takes place. So in this methotrexate, it has a N-methylation. That means first difference is N-methylation of the para-amino benzoic acid, and second difference is what? Replacement of this hydroxyl group of the pyridine with bioisosteric amino group. So both these are the bioisoster of each other, OH and NH2. Okay, these are classical bioisosters of each other. They are replaced, so OH is replaced with NH2. So what is the biggest significance of replacement of this? Just look there, The conversion of hydroxyl to amino increases the basicity of the N3 and yields greater enzyme affinity. So it increases the enzyme affinity. So both these drugs are the competitive inhibitors for a particular enzyme. So because of the presence of this amino group, this drug has better affinity for that particular enzyme. And what is that enzyme? That enzyme is nothing but your DHFR, that is dihydrofolate reductase. Okay, this drug competitively inhibits the binding of the substrate folic acid to the enzyme dihydrofolate reductase, resulting in the reduction in the synthesis of nucleic acid bases. Perhaps most importantly, the conversion of uridylate to the thymidylate as catalyzed by thymidylate synthetase. In addition, purine synthesis is inhibited because the N10 formyl tetrahydrofolic acid is a formyl donor involved in the purine synthesis. The interconversion of various folate species is also shown in this scheme. Okay, lastly we have seen that the tetrahydrofolates are the cofactors for the normal biosynthesis of purine. That also we have seen, right? So next, so what is the mechanism of action of this folate reductase or the folate antimetabolite, that is methotrexate? Is it the folate molecule is there, folate or folic acid is converted into a dihydrofolate and which then converted into a tetrahydrofolate. So this is the enzyme required, that is dihydrofolate reductase, for the conversion of folic acid into a dihydrofolate, right? So instead of converting this enzyme, this folic acid, instead of converting into this dihydrofolate, this methotrexate inhibit this enzyme dihydrofolate reductase. This binds with this enzyme and the binding of the methotrexate is increases. How it is increases? By addition of the or by replacement of the hydroxyl group with the amino group. So the affinity of the methotrexate to this enzyme will be more. So this enzyme will binds with the dihydrofolate reductase and inhibit this action, and the tetrahydrofolate will not be formed. And what is the use of that tetrahydrofolate? Just now we have seen that this tetrahydrofolate is used as a cofactor for the synthesis of the thymidine, and that thymidine is basic important molecule for the DNA synthesis. Okay, so everyone got this point? Okay, so this is the basic mechanism of action of this antimetabolite. You just remember one step or the one pathway for the synthesis of the DNA. There is a more complex formation takes place, or this biosynthesis of the DNA is having a very complex structure, and every enzyme, every step, various substrates are interrelated to each other. But we have just studied only a particular part, that is the action of this antimetabolite in that purine, pyrimidine and folate that is involved. So particular part we have studied. So just remember that part only. So the methotrexate is the broad spectrum antineoplastic agent which is commonly used in the treatment of acute lymphoblastic and myeloblastic leukemia and other lymphomas and sarcomas. The major side effects seen are bone marrow suppression, pulmonary fibrosis, gastrointestinal ulceration. The leucovorin is often given 6 to 24 hours after the methotrexate to prevent the long-term effects on the normal cell by preventing the inhibition of DNA synthesis. The related to this is Pimentrex, but its scope is greater in that it does not only inhibit DHFR but it also inhibits the thymidylate synthase and glycinamide ribonucleotide formyl transferase, that is GARFT, which is involved in the purine biosynthesis. The material is taken into the tumor cells by the reduced folate carrier system and subsequently is converted into a polyglutamate which is retained in the cell. Okay, so next, individual drugs. Just we will overlook on the individual drug. In that also I have explained the mechanism of action in short line. Okay, but the important part or the important schemes are there that I have already told you. So just revise the things by explanation during the explanation of this individual drug. The first drug is the 5-fluorouracil. From which class it is? It's a pyrimidine derivatives or pyrimidine analog. This drug is available in the 500 milligram to or the 10 ml vial for the intravenous use. It is also given in the form of the cream, that is 1% to 5% as a topical cream. This is used in the treatment of the several carcinoma type including the breast cancer, colorectal cancer, stomach cancer, pancreatic cancer, and the topical use is in the basal cell cancer of the skin. The mechanism of action includes inhibition of the enzyme thymidylate synthetase by the deoxyribose monophosphate metabolite, the 5-fluorodeoxyuridyl monophosphate. The triphosphate metabolite is incorporated into the DNA and the ribose triphosphate into the RNA. These incorporations into growing chain results in inhibition of synthesis as well as the function of DNA and RNA. So next drug is capecitabine. The drug which is available in the dose that is 150 milligram and the 500 milligram. It is available in the form of the tablet. This drug is the prodrug. Okay, that means it undergo activation and it undergo some metabolic step in our body and get the metabolite of this drug is the active form. So it's a prodrug. So what is the how it shows? That means the fluoropyrimidine carbamate. It forms a 5-fluorouracil. Okay, and the rest things are obviously similar to that of the 5-fluorouracil. It is used in the treatment of breast cancer, colorectal cancer. The drug is converted into 5-fluorouracil by the enzyme thymidine phosphorylase following stress activity to hydrolyze the carbamate moiety and deamination. So this is the steps required for the activation of this capecitabine drug. This capecitabine is readily absorbed in the GI tract. The peak plasma concentration is 2 hours after oral administration. Indications and drug interactions or all these related things are same to that of the 5-fluorouracil. The toxicities includes myelosuppression, leukopenia, thrombocytopenia, nausea, vomiting, anorexia, diarrhea, mucositis. The neurotoxicity is usually expressed as ataxia, lethargy, confusion, and allergic reactions often described in the pediatric patients include fever, skin rashes, conjunctivitis, and the chest pain. Next drug is the mercaptopurine, which is a purine analog. It is also referred as the 6-MP. Don't be confused that it's the 6 Madhya Pradesh, or it's a 6 mercaptopurine, 6-MP. Purine ethanol, purine ethanol. The drug is available in the dose of 50 milligram and it is available in the form of the tablet. This is a primary use. The primary uses of mercaptopurine are in the treatment of lymphoblastic leukemia, acute lymphocytic leukemia, Crohn's disease. The mechanism of action includes incorporation of this mercaptopurine into the DNA and RNA via triphosphate metabolite. This incorporation inhibits the synthesis as well as the function of the resulting modified DNA and RNA. The parent drug is inactive and requires phosphorylation for their activation. The resistance can occur via decreased expression of activating enzyme. So if this activating enzymes is not there in our body, then it shows the resistance. Then next, oral absorption is generally incomplete, about 50%. The drug does not enter the central nervous system. Mercaptopurine is metabolized by the methylation, and the methylated product has anti-cancer activity. So its methylated product, that is the its metabolic product, is also having a particular action. So it also metabolized by the oxidation into a by the enzyme xanthine oxidase, and which gives the inactive metabolite. So this drug is used along with the xanthine oxidase inhibitors. Why? Because it prevents the metabolism of this drug. When the xanthine oxidase inhibitors are given in combination with this mercaptopurine, the drug which is given, that is the xanthine oxidase inhibitors, they inhibit that xanthine oxidase enzyme, and it prevents the metabolism of mercaptopurine by oxidation pathway. So what is xanthine oxidase inhibitor? Example is allopurinol. It's a well-known and this is a well-known xanthine oxidase inhibitor drug, and this drug is enhances the potency of the mercaptopurine by inhibiting its catabolic breakdown. So the toxicities for the mercaptopurine include myelosuppression, immunosuppression, nausea, vomiting, diarrhea, dry skin, urticaria, and photosensitivity. So next drug is the methotrexate. This is a folate antimetabolite or the folate analog. This drug available in the dose of 50, 100, 200, 1000 milligrams in the vial. These are given intravenously. This methotrexate is used to treat the several cancer types, including the breast cancer, bladder cancer, colorectal cancer, head and neck cancer. The mechanism of action, it inhibits the DHFR, that is dihydrofolate reductase, which then leads to the depletion of the critical reduced folates, and reduced folates are necessary for the biosynthesis of several purines and pyrimidines. What is the resistance for this drug? The resistance is occurs because of decreased carrier mediated transport of the drug into the cell or increased expression of the target enzyme DHFR. So oral bioavailability varies with dose because the saturable uptake processes and high doses are required to reach the therapeutic level in the central nervous system. The majority of these drug doses is excreted unchanged in the urine. The renal excretion of methotrexate is inhibited by several carboxylic acid drugs such as penicillin, probenecid, non-steroidal anti-inflammatory agents, and aspirin. The methotrexate enhances the 5-fluorouracil anti-tumor effects when given 24 hours prior to the fluoropyrimidine. The methotrexate toxicity includes myelosuppression, mucositis, nausea, vomiting, severe headache, then renal toxicity, acute cerebral dysfunction, skin rashes, and hypopigmentation. So these are the different side effects of this ototoxic effects of this methotrexate. Then next is the pemetrexate. It is the MTA or Alimta. This drug is available in the dose of 100 milligram. It is in the sterile vial, given intravenously. Again, same. All the parts is similar to that of the methotrexate only. So it's used in the treatment of mesothelioma, mesothelioma, colorectal cancer, bladder cancer and lung cancer. The mechanism of action involves inhibition of thymidylate synthase. It also inhibits the thymidylate synthase, which results in the inhibition of thymidylate as well as DNA synthesis. This drug is pyrrolopyrimidine analog of the folate with antifolate activity. Resistance can occur by increased expression of thymidylate synthetase, decreased binding affinity for the thymidylate synthetase, or decreases the drug transport into the cell. This drug is administered only via intravenous route and these are distributed all the tissues. Cellular activation to the more potent polyglutamated forms occurs and the majority of the dose is excreted unchanged in the urine. The drug interaction and toxicity profile are similar to that of the methotrexate. The next drug is the trimethotrexate. This is available in the form of the lyophilized powder. This drug is given intravenously. The drug is used in the treatment of the colorectal cancer, head cancer, neck cancer as well as NSCLC. It means non-small cell lung cancer. The mechanism of action of this drug is the folate antagonism as well as inhibition of thymidylate synthetase. Trimethotrexate does not form intracellular polyglutamate adducts as does the methotrexate. Okay, the side effect of the methotrexate or the limitation of methotrexate is that, and other related compounds. The resistance can occur by increased expression of the target enzyme, the decreased binding affinity for the target enzyme, or decreased intracellular drug transport. Trimethotrexate is administered only intravenous route and it is distributed throughout the body with extensive binding to the plasma protein. The major catabolic pathway involve O-demethylation followed by glucuronide conjugation. So this drug is more effective one. So this is all about the antimetabolites. Okay, so various examples or all the mechanism of action we have seen, that is very important. Okay, now we will stop here. In the next lecture we will discuss about the antibiotics, that is antibiotics, neoplastic agents. Thank you.